This is Chapter 10, Enzymes, continued commentary. So now we're going to quickly review and then address more topics pertaining to enzymes. So in this instance, let me go ahead and pull out my red pen. So here I have my enzyme. The yellow is the active site. And just as a quick summary, it sweeps up and optimizes alignments for key biochemical reactions. So for example, the best analogy is like my marker. If I were to sit there and put a cap onto my marker, the best way to apply the least amount of force is to make sure the marker cap is parallel. If, however, I put it in at an angle, it either wouldn't go on right or it would require a lot more energy in order for it to go on. So enzymes optimize collision alignments between reactants and consequently lower the activation energy required for them to react. Now, the special name for any type of reactants which are going to be catalyzed are substrates. Now, in this part, we're going to be talking about inhibitors. And in a nutshell, what happens is inhibitors inhibit a enzyme from working properly. So it either stops or it decreases the enzyme activity. So for example, I could have an inhibitor and I could have it temporarily latch onto and block any type of incoming substrates. And with this, what we call a competitive inhibitor, as it blocks the active site, the substrates cannot obviously go into the active site. So it's blocked and then they have to go somewhere else. So whenever an inhibitor blocks the active site itself, we say it's a competitive inhibitor. Because the inhibitor and the substrate are competing against each other for the same active site. It could have been that even though the inhibitor was coming, all of a sudden the substrate cut off the inhibitor and was catalyzed at the active site. So whenever you have a substrate and inhibitor competing, we call this a competitive inhibitor. Now it turns out that there could be an alternate spot where inhibitors can dock onto the enzyme. I'm just going to highlight this with a green. This site we call a allosteric site. Allosteric means alternate site. So it's possible to sit there and have an inhibitor log on to the allosteric site, at which point notice how the inhibitor is not competing with the substrate originally shown. Instead, it has its own special pocket that it is able to dock into. Because it's not competing for the substrate, and just by locking onto it, it will inhibit the enzyme. We call this a non-competitive inhibitor. And our let's go ahead and go back to our enzyme inhibition slide. So it's pretty wordy here, but we're talking about inhibitors, and we have two types of inhibitors. We have reversible inhibitors and irreversible inhibitors. So in the example I just gave you, these are examples of reversible inhibitors, competitive and non-competitive. Let's go to a much better looking slide here. So for competitive and non-competitive, the inhibitors are always temporarily bound. The reason for this is the inhibitors allow us to slow down or even stop the enzyme, again, temporarily. But at a later point, the cell may want the enzyme to go to full enzyme activity. So therefore, it separates the inhibitors, or the inhibitors leave from the enzyme. So this is all temporary. What happens if you make it permanent? Well, if it's permanent, then we say it's an irreversible inhibitor. The body doesn't like those. Matter of fact, anytime you have an irreversible inhibitor, such as cyanide, or any type of heavy metal, such as lead, you can consider these outright poisons. Because once they bind permanently to the enzyme is dead. It no longer serves as any type of purpose. So you've killed, so to speak, the enzyme. Cyanide, for example, is a great example of a irreversible inhibitor. It actually locks on to oxidation reduction enzymes, which are responsible for making ATP. So if it kills those enzymes, you can't make the ATP, and then the cell basically runs out of portable power. So now we've covered irreversible, we covered reversible. I've gone into detail on competitive and non-competitive. Here is examples of competitive reversible enzymes. In this case, both the substrate and the inhibitor are both competing for the active site. Many type of drugs are competitive molecules. They're competitive inhibitors. So in this blown up example here we see a simulation of a new type of drug that was being developed called sucrose octasulfate. It is a competitive inhibitor because in that yellow box, which is the active site, it was actually docking to the active site and blocking this actual substrate. This substrate helps catalyze the wound healing process. So oftentimes we need to upset the wound healing process, for example, if a patient were in surgery. So this is a drug which basically docks and blocks the active site from the important substrate responsible for wound healing. This is a non-competitive inhibitor because they're not competing for the same place. The substrate still has full access to the active site. The inhibitor has its own special pocket, and when it bonds onto the allosteric site, basically what happens is it causes all of the side chain intermolecular forces to shift slightly, which disrupts the active site. However, the minute the non-competitive inhibitor leaves, then all of the temporary changes are undone, and then you have a fully functional active site again. Zymogens are responsible for doing a variety of events in the body. One event, they're used as digestive enzymes. Another one, they're used to break down fibrin and blood clots. So Zymogens are an interesting type of enzyme because normally they're permanently inactive, unless you have another enzyme who basically snips the tail of the inactive Zymogen enzyme. The minute you snip the tail, it automatically makes it active. So it goes from being permanently inactive, aka a apoenzyme, to permanently active, a holoenzyme. This is important, for example, with trypsinogen. Trypsinogen is the apoenzyme, and you wouldn't want these start to digest proteins within a cell. You want it to be delivered to the intestine. Once it's in the intestine, then you want it to start doing its job. So that's when you actually snip the tail, and then it will be a fully functional digestive enzyme which hydrolyzes proteins into amino acids. Plasminogen is another example. You only want it breaking down fibrin when you need to get rid of a blood clot. So you wouldn't want to get rid of a blood clot as soon as you cut yourself, for example, only after the body has a chance to heal. So know these two examples, know the names before, and know the names after. So before they're cut, plasminogen, trypsinogen. After they're cut, trypsin and plasmin. In general, we need to control chemical pathways. So in this example right here, in this example right here, here is one way that we can control a chemical pathway. So a chemical pathway is a series of chemical reactions where A will be changed to form product B, and B will be changed to form product C, care of important enzymes along the way. What happens is, one way that we can control enzymes is that some of the future products, they can act as inhibitors. So, feedback inhibition is an example of a modulator decreasing an allosteric enzyme's activity. So notice how I jumped right into this. I don't want you to get caught up on this term modulator. Modulator basically means how are you affecting the enzyme. If you're increasing the enzyme activity via an allosteric site, we say it's an activator. If you're decreasing it, then we say that it's a non-competitive inhibitor. So don't get thrown off on these terms. They're very easy terms. Again, if you're increasing it via an allosteric site, it's an activator. If you decrease it, then it's a non-competitive inhibitor. So let's see an example of feedback inhibition. This right here is a enzyme. And again, we're looking at a pathway where A will sit there and create a product B. That's going to be used as a substrate, and another enzyme-induced activity to create C, and so forth. Notice down the line we have this product that's formed called isoleucine. This isoleucine is an example of a non-competitive inhibitor. So one general example of a modulator which decreases this particular enzyme's activity. So what happens is this will sit there, and isoleucine will, as we increase in concentration, we need a way of cutting down the amount of products that are produced because maybe they're not being used. So one way to do that is to take those excess products and have them being used as non-competitive inhibitors. This then slows down the enzyme and therefore slows down the production. The minute, however, we need extra amounts of isoleucine because we're running low on our product, then there's going to be less of this non-competitive inhibitor floating around, so therefore most threonine deaminase enzymes will be at optimal enzyme activity. Medical applications. Here is a in-depth example of a person's heart. So there's his chest, there's the manly hair if it's a guy. Hope it's a guy. Yeah, manly hair from a guy. And the doctor is wondering, did he have a heart attack? It turns out that whenever cells are damaged, many cells will sit there and die, and then they will actually rupture. And they will release all of their enzymes within the cell. So, for example, a popular enzyme found in heart cells is one called creatine kinase, and we'll summarize it as CK. If a patient were to have a heart attack, then many of these cells would rupture, releasing creatine kinase into the bloodstream. So this is my high-dev example of a artery, and basically it's found in your bloodstream. Consequently, when doctors want to know if you've had a heart attack, what they can do is they can go ahead and check your blood to see if you have high concentrations of these key enzymes. If you do, then it confirms that a heart attack, for example, might have occurred. If you don't, then the doctor will probably do additional tests. So this is one way that doctors can figure out what's wrong with you. They know that whenever cells are damaged, they may release key enzymes in the blood. So if you ever go to the hospital, oftentimes they may say, okay, we need to run a blood test. And now you know why. They're looking for increased elevated levels of enzyme. Know these different types of Be familiar with these different types of enzymes. Knew them. Although I may not ask you to list them all, I may ask that you give me an example, such as the one I just gave you. For example, they may be looking for creatine kinase, or they may be looking for creatine kinase and aspartate transaminase.